Hello, everyone. I welcome you all to the webinar on Winter Research Fellowship Program. This Research Fellowship Program has been designed to help young researchers and students take advantage of the bioinformatics resources for analysis of complex high-throughput life science data and become versed in bioinformatics. This program offers you to participate in real and cutting-edge bioinformatics research and to be a part of renowned bioinformatics community. OmicsLogic is an international program that is running in 5 different regions with over 15,000 users around the world. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. OmicsLogic programs follow a project-based learning approach using research-grade tools to analyze data from top peer-reviewed journals. And also, the training has been completed by over 10,000 participants from 162 countries in over 300 workshops for 6 different specialization tracks, being oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Our training is designed to allow participants to apply methods to curated and new data sets and develop independent projects that showcase what has been learned in the context of a personal portfolio of projects. As a result, anyone starting on this journey in bioinformatics can start developing a project of their own. In fact, we have already worked with many students who have completed this kind of training and developed interesting research projects that won awards and got published in peer-reviewed journals as well. In this session, you will hear about some of the critical points that makes a project ready for a peer review. We will discuss the structure of the program and also hear about network of campuses, expert mentors. And we will also hear updates from students and mentors who are supposed to complete the projects and learn about various resources that are covered in the program. The resources that are covered in the program includes the OmicsLogic courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics as well. Data science that includes bioinformatics using R and Python, and the structural aspects of biology, that is cheminformatics. As a part of the program, you would also get the access to the example projects from different fields, which you could learn from and replicate for your own research work. You can also explore various projects by our research fellows in various fields, including neurosciences, genomics, transcriptomics, infectious diseases, etc. Now, I would like to pass on the stage to our mentors for further discussion about the program. And at first, I would like to invite Ilya Brodsky, CEO, Pine Biotech, to talk more about the structure of the program and why is it really important for you all to work on a research project. Over to you, Ilya.
Thank you very much, Sonarika. Share screen. So it's exciting to see many of you join today. And I think that many of you are probably wondering what makes this program different or special in the available programs online where you can learn about bioinformatics, you can learn about data science. The reason why we developed this program is specifically to go beyond training and offer an opportunity to participate in meaningful research. Now, in today's session, we're going to address many of the challenges that are associated with research projects, especially getting those research projects completed in a short amount of time, because typically they do take years, at the minimum, several months. And so what I want to talk to you today about are some of the critical things that we've distilled into this online program and introduce you to some of the mentors to talk about some of the specific project examples. But before we do that, let me briefly address some of these critical points. One of the things that we've realized is that when people join our different training programs Their background in life sciences might prevent them from really getting deeper into bioinformatics methods, allowing them to superficially know the terminology, but not really know how to apply it practically. So I think that, many of you will discover that some of the training materials that we've designed are specifically useful for those that come from a life science background without a lot of technical engineering skills. And then you will be able to really appreciate both the logic of the analysis and technically how to become independent and efficient in bioinformatics analysis methods. The second area where many students face a challenge is finding an impactful research topic. And to find that impactful and meaningful topic, we've addressed this through two different approaches. The first one is literature review. How to perform literature review so that you can find the specific gaps in research and opportunities for you to do something new and different, and at the same time find data that you can find answers for that question, that research question. So it's developing the research question, finding the data. We review many data sets with our community to provide you with a curated start so that you can start investigating a topic using a curated data set and then find opportunities to innovate and ask questions of these data sets, including introducing you to the approach of finding data sets online. And then finally, the third topic is how to know that what you have developed is a meaningful project, not just for the student community, but also for the scientific community. And that's why we have developed a network of mentors that come from a research as well as industry background that are able to provide you with critical feedback so that your project becomes more impactful and refine the project, or provide you with suggestions on how to refine the project to make it more impactful and essentially providing you with more visibility and feedback. So before we continue, I want to show you how some of these things are going to actually take place. And I want to introduce you to some of the program resources really briefly. And to do that, I want to point you to two servers and two portals. One of them is going to be the server T-BioInfo portal. This is going to be a portal that is designed to provide you with an opportunity to process big data, and also the Learn OmicsLogic portal, where you will find all of the training resources. So let's first go to this portal, the OmicsLogic portal, and thank you, Ajasvi, for sharing that link. And I'll also explain how those two things are linked. So if you go to this portal link, you will find here, I think, a very clear explanation of what this portal really is. The OmicsLogic portal is designed to provide you with everything to get you started and get you into these real world practical challenges with big data. And you will find here tutorials, project examples on how to run various pipelines and apply different analytical methods. You will find here different types of research projects that you can get started with in the fields of oncology, infectious diseases, neuroscience, agriculture, and others. And you will also find meaningful application of your logic. How do you understand that you can take raw data and get structured data that you can analyze and supplement that with training with coding and machine learning and data science and analytics, all of the necessary skills that will make you independent and self-sufficient as you go forward with your projects. So the first thing that you want to do is you want to click right here on login, and I will give you a couple of minutes to complete your registration. So once you click on login, you have the option here to create an account. Okay, so I will wait a few minutes. For those of you that don't have an account, please take a few moments right now to fill in your name, your email, and create a password. It's a free account. You don't have to pay for this, but you will immediately get access to some of the free resources that are made available, and hopefully they will inspire you to continue forward with a research project using bioinformatics. So once you have completed your registration, please share on the chat box the link to your profile. And I will explain briefly what you can do with this profile. Okay, so I will show you my profile. Okay, once you log in, you can click right here and you will find your profile open up. And so if you want to share your profile, and just go right here to the URL on top, copy this URL, and you can share this right here with everyone in the chat box. So please let me know. I'll give it a few minutes so that at least a few of you will be able to complete your profile and share it with us. For those of you that already have an account, thank you, Aranza. For those of you that already have an account, especially the campus ambassadors that are here in this session, please share your accounts as well. So we will take some account that is complete so that we can show how a completed profile looks. Okay, thank you. All right, great. So let's take just one of these accounts as an example. Here we have an account by Subrajit, who is already an expert. How do we know that he is an expert? He's an expert, first of all, because of the activity that we can already see on his profile. So you can see that all of your activity as you go through the lessons, as you try different projects, as you try different analysis techniques are quantified. And what that means is that you get a number of points for completing each step, and that immediately signals to the mentor community that you have accomplished the basic training and you're ready to get started with your research project. So the way the research fellowship works is it starts from this basic training. What is covered in the basic training? So importantly, I think in today's world, when we speak about bioinformatics, the major focus is on big data. This big data comes in the form of genomics, transcriptomics, metagenomics, epigenomics. There are many different types of omics, and ultimately you have to know how to process each individual types of data and then integrate them together to really have this systems level view of what's happening in a biological condition. And so we've designed these so that you have an introduction, and I will show you a couple of courses that you can start with for free so that you get into this whole field of omics data. Then you go through this basic training, and then you get introduced to the data science curriculum in R and Python. So I will briefly show you a couple examples, and this is just to get you started. So again, as you can see this profile, it is complete with activity. I can see which courses Subrajit has completed, and I can also see that he has earned certificates, and those certificates are going to stay with you unless the course has been updated. And when it's updated, your certificate will be taken away so that you can complete the remaining lessons and then get the certificate back. And that way, we know that you're staying up to date on some of these important topics. In addition to that, you can participate in programs. And today we're speaking about the Research Fellowship Program, but also we have many others that are led by mentors who are experts in the field. And you will also be able to develop a project. And so as we go through, one of the requirements that this program has is so that you complete your basic training. Now, where would you start your basic training? Here on the courses tab, right here in the menu, you can find a lot of courses that as you work through some of the material will get a personalized suggestion for your profile. So you can see that for me, I have some suggestions on single cell transcriptomics, machine learning, Ebola virus, etc. And that's because of my interests. But here below, I have a list of recommended and highly active courses, as well as some of the collections of courses that you can find right here. And one of the collections is called Getting Started. So I can just click right here on Getting Started and see what's included. The course that we recommend for all of you to start with is a course called Bytes and Molecules. The course explains many of the basic terms. Sorry. A lot of the basic terms that you need to know to get into this field, and it is using language that anyone can understand, both with and without the kind of bioinformatics background, and it covers important topics that you will want to understand before you really get into a lot of the material that is related to research using bioinformatics. So that's just where you would want to start with. So after you complete enough material to generate 10,000 points, what will be made available is right here under projects. You will be able to start a new project. And that is essentially what this program is all about. So this program is going to be focused on getting beyond the training after you've completed the 10,000 points and into a research topic. Now, one of the immediate challenges with that is how do you process this huge amount of data that oftentimes is beyond several gigabytes or even terabytes of data that you have to download on your computer. And so for that, we specifically designed a big data analysis platform that will cover many of the typical processing steps that reduce the amount of data from gigabytes and terabytes to just megabytes, which is possible to analyze either on your system or in the cloud. And we will introduce you to cloud technologies using this platform called the T-BioInfo platform, developed at the Tauber Bioinformatics Research Center at University of Haifa, as well as some of the cloud technologies offered by other companies like Google Colab, GitHub, and other resources that are going to be useful for you to leverage as you develop your career in bioinformatics. So here we have, I'll just briefly show you one example. As you probably all know, transcriptomic data on RNA sequencing is becoming more and more prominent in many different areas of research, including oncology, neuroscience, and other fields. And briefly, you can appreciate the simplicity of this platform because it really organizes the information in a logical, step-by-step process. And by the way, this is reflected in the coursework that you will find on the Learn portal. It also guides you using artificial intelligence based on the data that you upload and the information that you would like to receive from this pipeline so that you can eliminate the common mistakes that people make when they try to design these workflows on their own. Now, as a result of running these workflows, you also get a clear sense of the logic behind the analysis, including the inputs and outputs of different methods. And so once you're ready to transition to your own independent pipeline that you want to build on your own, now you at least understand the logic. Why would I use this method and not that method? When would I try to modify this setting or that setting? And this spans, as I mentioned, across many different areas of analysis, including next generation sequencing data, mass spectroscopy, structural biology, as well as many methods for data integration and modeling, machine learning, and other types of applications. So all of you can definitely go through some of these resources. The thing is, once you've created an account on the OmicsLogic portal, you automatically get access to the demo level on this bioinformatics platform after 24 hours. So from the moment that you created a profile today, tomorrow morning, you can already start using this platform, seeing how it is used in different courses and projects that we will talk about. So this is the T-BioInfo platform. Now, ultimately, you're probably wondering, well, okay, I have these resources. I can go through the training. I can understand what topic I would like to analyze. I can perform the analysis, but how do I know that I'm doing the right thing? And so to do that, we have a community of mentors. And I think that's a very critical point to briefly discuss. So our community of mentors, first of all, is an international community. Many of our mentors come from research backgrounds. Many of them have a PhD in a particular domain like bioinformatics, computational biology, neuroscience, oncology, etc. And today you will hear from some of them. but also many of them come with industry experience. And so for those of you that understand what you want to do with your career, if you would like to go beyond basic research and focus on translational research projects, that is one of the specific goals of this program as well. As a research fellow, you get to explore a variety of topics and select one that is either relevant for industry or for research. And we'll talk about this today, including different collaborations that we have with universities and organizations that provide us with this opportunity to know what is needed and is in demand at the cutting edge of basic research and industry R&D as well. So what are some of the outcomes? The outcomes of this program is that you will come out knowing how to apply bioinformatics to a particular project, and you will be able to complete that project within a short period of time. And just to give you a sense of what I mean by that, I want to pass this presentation now to Dr. Harpreet Kaur. She is a mentor with a background in genomics for oncology research. Has a lot of expertise working on many different research publications together with her collaborators, but now she's a mentor and she has been working with several research fellows over the past couple months that will present their updates on projects so you can get a sense of where they are and how they got to where they are. So Dr. Harpreet, I will pass it on to you now.
Thank you, Ilya. Let me share my screen. Hello everyone, myself Harpreet and I have a PhD in bioinformatics. In my current role, I am curriculum developer and research consultant. I am mentoring number of research fellows and working with international collaborators on various research projects based on biomedical NGS data. So today we will also see some of the research fellows who are presenting their progress on their respective projects. But before their presentation, let me give you a glimpse of how bioinformatics can be used on biomedical data to drive significance, meaning by taking some example from the projects that I have completed during my doctoral degree and some of the projects that we have completed during this research fellowship program. So my PhD is in the area of cancer genomics, specifically liver cancer. So when I went through the literature, I found some of the major challenges associated with the liver cancer, including liver cancer is often misdiagnosed or detected too late for an effective treatment. Another factor that contributes to the poor outcome for the liver cancer patient is the frequent reoccurrence of the tumor after the treatments. So keeping these concerns in mind, so we have worked on several projects. So in the first project, we used large-scale transcriptomics data to identify a diagnostic biomarker panel for hepatocellular carcinoma. In this study, we employed 30 datasets containing nearly 4,000 samples, and this data was taken from public repositories like GEO and TCGA to elucidate transcriptomics biomarker panel for the diagnosis and the prognosis of the liver cancer, implementing various statistical and machine learning techniques. For the next project, we used transcriptomics data to scrutinize the diagnostic biomarker panel for cholangiocarcinoma, which is another liver malignancy. In this study, we employed 9 datasets from public repository, mainly GEO, which include 350 cholangiocarcinoma, 133 adjacent non-tumorous and 90 HCC sample to scrutinize cholangiocarcinoma specific transcriptomics biomarkers, implementing various supervised machine learning techniques like SVM, random forestic, logistic regression, etc. So in the next part, we focused on the prognosis of liver cancer patients. So here in this project, we used transcriptomics and methylation data of hepatocellular carcinoma to identify biomarker panel that can classify early and the late stage of liver cancer. So in this study, we have again taken public data from TCGA repository, which contain early and late tumor stage samples. So in this study, we identified 51 signatures, which include 30 RNA transcripts and 21 methylation CpG sites, using a number of machine learning and statistical approaches. So in the last part, we focused on identification of multi-omic signature that can stratify low reoccurrence risk group and high reoccurrence risk group of SCC patients. So in this study, we identified 90 RNA, 50 miRNA, 50 methylation, and 4 integrative features that can able to significantly stratify reoccurrence risk group of SCC patients. So now let me give you the glimpse of some of the projects that completed by a research fellow in this OmicsLogic Research Fellowship Program who have not much background in the bioinformatics. So we have research fellows from various backgrounds and across the globe. So here one of the research fellow, Ms. Simay Dolanar, who is an undergraduate student from the Turkey, completed her research project entitled Long non-coding RNA as a therapeutic target in chronic lymphocytic leukemia using publicly available data in employing various bioinformatics tools from T-BioInfo Server, which Ilya has just shown you. Then Ms. Urja Parekh, who worked on the project entitled Identification of Biomarker to Differentiate between Neural Stem Cell and Glioblastoma Stem Cell, employing publicly available data using various data mining techniques. So today, Ms. Urja Parekh is also going to present her progress on the project. Then, apart from the oncology projects, so then we also worked on various other areas, including neuroscience. So here, our one of the research fellow, Ms. Rutvi Vaja, completed her project in the field of multiple sclerosis. So she worked in the project entitled In Silico Analysis of Transcriptomic Profiling and Affected Biological Pathway in Multiple Sclerosis using publicly available data. Then beside, some of the research fellows also worked in the area of spaceomics. So for example, here, Ms. Dilara Dekane completed her project in the field of spaceomics entitled Radiation 3 to Humans in Space, an alternative approach with probiotics using metagenomics data and various bioinformatics tools from T-BioInfo Server. Then beside oncology and neuroscience, then we also worked in the field of infectious disease. For example, Mr. Supantha Dey completed his project in the field of infectious disease, particularly malaria. So he worked on a project entitled Analysis of Gene Expression Profile to study malaria vaccine dose efficacy and immune response modulation using transcriptomics data of the patients and various data mining techniques. Then another example of infectious disease project that we have completed. So we completed with our collaborators from Ghana on the project where we have tried to identify haplotype variants of the Plasmodium falciparum AMA1 analysis based on the genomics data. So then apart from masters or undergraduate students, we also worked with the high school students. So let me give you the glimpse of the projects that completed by high school research fellows. So here Ms. Netra Srinivasan completed her project in the field of neuroscience. She worked on the project entitled Effect of Onset of Menopause on the Transcriptomic Profiling of Aging Female Brains. Besides, another research fellow, Mr. Andres, completed his project in the field of Alzheimer's disease. So my major research interest lies in cancer genomics, infectious disease, neuroscience, and machine learning. And specifically, my focus is most of the studies for the biomarker discovery. I'm looking forward to work with many of you. Now, I would like to pass this session to Dr. Mohit Majumdar to talk about other research fellows. But before his presentation, let me invite some of the research fellows who are currently working on various projects. So first, I would like to invite one of our research fellow, Mr. Aviu. So title of his presentation is Identification, Characterization and Analysis of cell wall related genes as primary target for efficient biofuel production in sorghum bicolor. So over to you, Aviu, so you can share your progress update. So Aviu, are you here? So I think he might be get disconnected. So now I would like to invite our another research fellow, Ms. Urja Parekh. So title of her presentation is In Silico Analysis and Characterization of Differentially Expressed Genes to Distinguish Glioma Stem Cells from normal neural stem cells. So over to you, Urja. So Urja, you need to unmute yourself as well. Okay, so Sonalika, can you just make co-host to Urja?
Yeah. Okay. Yeah.
Yes,
Yeah.
Susha.
Okay.
I hope I'm audible.
Yes.
All right. I'll share my screen. All right. Thank you very much, Dr. Harpreet, for introducing me. I'm going to be talking about my project in silico analysis and characterization of differentially expressed genes to distinguish glioma stem cells from normal neural stem cells. So a little about myself. I'll start my PhD in cancer biology with the German Cancer Research Center in Heidelberg, Germany. I have pursued my master's and bachelor's in biomedical science from NMIMS University, which is in Mumbai. And I've also topped my university for the same course. So with Pine Biotech, I have been working on this particular project on glioblastoma, where I am trying to elucidate the differences in gene expression between normal neural stem cells and glioma stem cells. So glioblastoma multiforme is a very lethal type of brain cancer where patients are given an overall survival period of only 15 months post-therapy. The reason why this particular brain cancer is so aggressive is because of its heterogeneity, and there are different types of tumors with different types of expression profiles. So we also currently do not know from where this particular type of cancer originates, whether it originates from normal neural stem cells via transformation of the normal neural stem cell type to the cancerous phenotype, or do they originate directly via the glioma stem cells. To get a better understanding of the same, I have performed RNA sequence analysis. So what I did was I looked at a current repository of RNA sequence for glioma stem cells and normal neural stem cells, from which in my previous analysis I've taken 40 samples, 20 of normal neural stem cells and 20 of glioma stem cells. And after performing RNA sequence analysis, I got results where I was able to see a clear separation between the glioma stem cells and normal neural stem cells in my PCA analysis. I was also able to observe a clear separation in the hierarchical clustering. I was also able to observe several differentially expressed genes. So 192 genes that were significantly upregulated in glioma stem cells and 156 downregulated genes in glioma stem cells in comparison to normal neural stem cells. So these particular genes are of importance because they might be playing an important role in driving gliomagenesis. So in order to better understand its involvement in different pathways, I performed gene ontology studies using David and KEGG, which are tools for doing such sort of studies, where I was able to find the involvement of these particular differentially expressed genes in pathways that are hallmarks of cancer or tumorigenesis, like Wnt signaling pathway, TGF-beta pathway, VEGF signaling pathways. So with literature review of the top 50 significantly differentially expressed genes, I was able to find that there are several genes that are playing key roles in this particular type of cancer. And there were several novel genes with currently no known association with gliomagenesis. Now, the reason that I'm extending the study is because I had taken a very small sample size, and I believe that a larger sample size is required to get more relevant findings. And I can also not generalize these particular results with other types of subsets of glioblastomas. And therefore, for the same reason, I am extending the study to perform something called internal validation and external validation. Now, I have taken the smaller data set, which I've already analyzed as my internal validation data set. I am repeating my study for 148 samples of normal neural stem cells and glioma stem cells. And I will also be performing external validation for which I'll be finding another type of normal neural stem cell sample and glioma stem cell sample and correlate the findings of my current study with that other study. So in the current study where I've taken 148 samples of normal neural stem cells and glioma stem cells, I have performed RNA sequence analysis. I have received gene counts after performing that particular pipeline. I have also performed differential gene expression analysis using DSeq2. And eventually I have performed gene ontology studies, human gauge pipelines in GC to better understand the involvement of genes enriched in my particular samples and how they might be driving this particular type of cancer. So the first PCA that I performed for the 148 samples that I've considered in the study, you can see that I got two separate clusters for glioma stem cells and normal neural stem cells. However, there were certain outliers, which were normal neural stem cell samples clustered with glioma stem cell samples. So in my further PCAs, I removed those particular outliers and the excessive genes that may not be of any relevance. And then finally, I got a clear and separate PCA showing the two different types of stem cell samples, as you can see in the third PCA plot. I also performed hierarchical clustering. Currently, the plot looks a bit messy, but it basically shows two clusters, one where glioma stem cell samples are and one where the normal neural stem cell samples are. I have also performed differential gene expression analysis using DSeq2, where I initially obtained 19,453 differentially expressed genes. However, on putting certain filters, such as the p-adjusted value, I finally got a p-adjusted value of lesser than 0.05. I got 3,977 significantly differentially expressed genes. I later also put certain thresholds on the log2 fold change values, which was plus 1.5 for upregulated genes and lesser than minus 1.5 for downregulated genes. So I got even fewer genes that are more significantly expressed in these particular types of glioma stem cells in comparison to normal neural stem cells. So I had this massive gene list, but I had to also interpret what these particular genes mean in terms of gliomagenesis, for which I performed downstream analysis. So I will be showing you all what exactly I've done using those particular softwares. But basically, I've used David and Enricher, which are gene ontology tools where you can insert gene sets and try and analyze where those particular genes are involved in, what functions are they involved in. I have also used the human gauge pipeline to elucidate the most significant pathways in which my genes are enriched in. And again, GC is something, it's gene set enrichment analysis, again, which gives us genes that may be correlated with current hallmark gene sets. So like, for example, we already have certain gene sets that may be involved in the TGF beta signaling pathway, and there are gene lists that have already been made by other scientists. So basically correlating my gene set with those known gene sets and seeing if there is any sort of correlation between them. So I will just stop my presentation here and show you all my results. So first I will show you all my gauge results, where, okay. Can you all see my main screen, or are you all still able to see my presentation?
Pooja, I think you need to unshare your screen first and then reshare so that we can see your transition to the other screen.
All right.
Yeah, thank you.
Yeah, okay. I hope you can see it now.
Yes, we can see. Thank you.
So these are basically KEGG pathways. So you can see genes that are highlighted in green and red. So these particular genes were present in my gene list, and they are basically drivers of gliomagenesis. So you can see that they are involved in pathways like the calcium signaling pathway or the axon guidance pathways or the focal adhesion pathway. So basically, there are different pathways that have been highlighted by the gauge pipeline, which essentially states that these particular genes are involved in these particular pathways. So I need to interpret this data further in order to elucidate how this might be correlated to gliomagenesis. So for example, we know that ECM receptor interaction is important for tumorigenesis. It gets altered when cancer when a person has cancer. So the cells have an altered ECM receptor interaction. So this further needs to be interpreted, which I'll be doing. You can also see its involvement as cell adhesion molecules and pathways that may be responsible for cell adhesion. So these are some of the pathways that I've got after the human gauge pipeline. Apart from that, I have also performed gene set enrichment analysis using the GC software. Again, I have got very large results for the same. Yeah, so for example, this is a heat map of a particular gene set. It's a hallmark gene set. For epithelial to mesenchymal transition. So EMT is a pathway that occurs during tumorigenesis, and there are certain genes that are highly represented in cancer overall. So these particular red and blue dots is what you can see are the genes that are expressed in my particular gene set as well. And red means that it is upregulated, and blue means it's downregulated. So on the left-hand side of this heat map, you can see that there are glioma stem cells, and on the right-hand side in yellow are the normal neural stem cells. So you can see that there are certain genes that are upregulated in glioma stem cells and downregulated in normal neural stem cells. This is a more in-depth plot of several genes that are upregulated in glioma stem cells and downregulated in normal neural stem cells. And again, there are certain genes that are upregulated in normal neural stem cells, but downregulated in glioma stem cells. So it kind of shows the differences of gene expression in both these types of stem cell samples, and they differ quite a lot. The other interesting finding I got from my GC analysis is so there are different types of subtypes of glioblastoma. So there is a mesenchymal subtype or a proneural subtype. So I have got different plots, heat maps for the mesenchymal glioblastoma subtype, as well as the proneural subtype. So I again need to like classify what kind of whether the neural stem cells that I have taken into consideration, what kind of subtype do they fall under for the glioblastoma tumor classification. So there is a lot to look at because I have used several different types of gene sets from GC to try and understand the gene sets that might be enriched in my particular study. So yeah, this is just like a general heat map of my particular study. Apart from that, I have also used Enricher and David. So I have put in the genes that are upregulated in my particular data set, and I have uploaded them on Enricher. So on Enricher, basically, I can see what kind of transcription pathways or ontologies or diseases my particular gene set might be enriched in. So currently, because there are so many results, I'm still trying to cut down on the most relevant or most important results and try and elucidate how my gene sets might be involved in these particular pathways that are shown over here. So I do kind of need help with that. So yeah, currently I am on the part where I am still trying to interpret the meaning of the thousands of genes that I have got out of my RNA-seq analysis or differential gene expression analysis. And what I further intend to do is compare these results with the results that I have got from the 40 samples that I previously analyzed, and also compare these particular results with an external data set so that I can say that these particular results that I have got can be generalized, and these might be important drivers of gliomagenesis overall. So I hope you all have understood what I have been trying to do, and I would love to get some feedback on the same.
Thank you, Urja, for sharing wonderful presentation. And yeah, for the pathway analysis, you can go, analyze it first, like what are the important specifically based on log2 fold chains and the p-adjusted value, you can find what are the important pathways, because you have a lot of lists. So just go into the table where you have pathway or a gene ontology term corresponding p or p-adjusted value. So try to select only important pathways, at least p-adjusted value less than 0.05. I think then you can get important pathways. Yeah.
Okay.
Okay. So now I would ask Ilya and Dr. Raghavendran to give comments on your presentation or any suggestion that they have.
Yeah, thank you. I think overall, it's clear that you've done a lot of work, and now the task ahead is to integrate all of these analysis results and kind of make sense of what are the clear findings. I think because of the time constraints, this is an exciting update to the project that you've been working on for a while. And you're obviously addressing a lot of the issues that came up during the initial analysis. Let's move on to hear a little bit from Dr. Raghav, who is another mentor, and then he can transition to his part of the presentation. So thank you, Urja.
Thank you, Elia. I think I'm audible. So that was a very detailed and very good work, Urja. And I'll be interested to see. Okay, it is not there. Yes. Okay, great. So I'm very much interested to see the pathway associated with it, and then you can do selected analysis on the pathway that are shared or that are not shared, completely different between the normal stem cells and the glioma stem cells. So those are very interesting ones. Maybe a even more stringent p-value could give you a reduced set of genes, because you still have a lot of genes to analyze, and that would, yeah. So you can maybe you can reduce your analysis or downstream analysis to top 20 or top 15 upregulated genes and top 20 downregulated genes for each set, and then try to see if these top 20 genes are from multiple pathways or the pathways associated with these top 20 genes. That should narrow it down and make your analysis even more workable, because I mean, at the moment you have really a lot of data to comprehend with. So right now I would start with that, and then and see where it goes, and maybe extend it to top 50 if that makes even more meaningful, even more meaning, and we will see. Yeah.
All right. Thank
okay,
you.
So thank you. Cheers. So I think that will translate or transition to my talk or presentation. I will share my screen right away. I just need to find it where. Okay, got it. Okay, excellent. Okay, that worked. Okay, great. Can everybody see my screen? I think you can. Yeah.
Yes, sir, we can. Thank you.
So yeah, thank you. I am Raghavendran, bioinformatics mentor and research consultant with OmicsLogic. Let me brief you about my my background and research interests. So I grew up to be a tinkerer kid who is interested in solving many problems. So right in my younger ages, computers and programming with computers introduced me to very new puzzles and challenges. So attempting to solve these puzzles and these challenges were the first step that took me towards solving puzzles and asking questions. So during my undergrad education, I read about the potential and the announcement of the Human Genome Project. And that was the first research article that I read. I didn't understand it, most of it, but it inspired me to a great deal, and I pursued my master's in biophysics. So my natural inclination towards computers and structural biology, bioinformatics, led me to specialization in computational biology and bioinformatics. So after my master's in bioinformatics or biophysics, I joined a PhD in Max Planck Institute for biophysical chemistry with a specialization in computational biology. So during this tenure, I developed software and designed a lot of methodologies and pipelines for protein structural and dynamic studies. So my system of interest during during the PhD period was proteins associated with neurodegenerative diseases. So at the time when the next-gen sequencing technologies were starting to get established in India, I came back to India as a postdoc to the Institute of Bioinformatics and Applied Biotechnology in Bangalore. The potential of the next-gen sequencing technique and its application to multi-omics data analysis, these two combined and piqued my interest in cancer. So you all know cancer is the leading cause of disease death worldwide. So I got naturally inspired and interested to work with cancer biology. So I contacted Professor Shandar Ahmed in JNU and Dr. Pedro Ballester in collaboration with Dr. Pedro Ballester in INSERM, France. I proposed a study to understand and to explore tumor responses to different treatment regimes. So this was while I was in Bangalore, and I also agreed to help a friend who was in need of bioinformatics help in de novo genome assembly of a variety of neem plant. So it was only during this collaborative project I came to realize how little information was available for plant bioinformatics in the public repositories. While when you take or when you compare it with the availability of databases and the repositories for human or bacteria, there is hardly anything for the plants. But there is so much potential to understand plant genetics, because through bioinformatics and through sequencing techniques, we can understand the underlying biology of plant genetics, because agricultural crops have been subjected to artificial selection for ages, irrespective of geographical location and irrespective of culture. So I continue to work with the project, and they are writing up the paper, and I'm still associated with the department for some troubleshooting in the pipeline that I developed. So while all this was going on, the project that we proposed was successfully funded, and so I joined JNU as a postdoc research scientist. So I will detail more about the data that I use for projects and about the projects and the results in a short. But I just wanted to mention, right from my PhD years, I enjoyed working with undergrad students and enjoyed working with students. So this experience tremendously inspires me, and I feel privileged to work with some of the bright young minds. So I hope and I wish every one of you will enjoy working with me, and as much as I look forward to working with you. So for the next two slides, I want to move on to speak about the data that I use for the project on precision oncology. Let's see if I can get this rolling. Yes, okay, great. So every once in a while in science, a groundbreaking research gets published, and the data collected for that work contains a lot of potential to answer a wide variety of scientific questions and queries. So this is one such paper, and this paper is about tumor cells that are grafted into mouse models to study tumor responses to different treatment regimes. So what they did was they extracted the tumor cells from cancer patients, and then they sequenced it, and from the sequence they profiled the gene expression levels and the gene mutation levels and the copy number variation levels, And then these tumor cells are grafted into immunosuppressant mouse. So, and these mouse were administered different and different set of treatment regimes, and the tumor's response to these treatment regimes were studied. And such mouse or such immunosuppressant mouse which got administered with tumor grafts are known as patient-derived xenografts. So these PDXs, in short, and they are administered 60 different treatments, and their response were recorded in change in volume and the ability of the mouse to survive the disease. So such survival data analysis of 1075 PDX mouse was released along with this data, along with the study. So this study from this analysis concludes that the mouse models provide a better alternative compared to the cell lines which were traditionally used, because cell lines are often affected by the petri dishes they are grown on, and heterogeneity is one of the biggest problem in cell line data so far. And at the basic genomic level, compared to the cell lines, the genes, the gene expression levels of the PDXs resemble more to the cancer patients than the cell line data. And PDXs are also consistent, or the data from these PDXs are also consistent with extensively studied therapies, and this comparison validates their applicability to understand the underlying cancer biology. So such a wealth of data generates a plethora of research questions, as I already posed. So one can employ this data to various analytical pipelines to study treatment stratification, like can the mutations in genomic data predict tumor resistance, or vice versa? Can tumor resistance relate to significant mutations in those models? Or are combination of drugs more effective than single drugs? Or how does different combinations of drug work well or does not work well together? Or can we find the underlying biology behind this? Or we can also do some precision medicine study, like do targeted therapy exhibit higher efficacy than general cytotoxic treatments? Because among these 60 treatments, some were general cytotoxic treatments which are toxic to all cells, just they kill cancer cells more than the normal cells. And some specifically target pathways inside the cancer cells. So can we find out the underlying differences between these two treatments? And with the help of multi-omics integration, can the PDXs broadly represent the interpatient diversity? Because that was one of the biggest conclusion of this study, where they observed a diversity among the models which carry the same cancer cells from different patients. So they translate into interpatient diversity. So can we quantify this diversity? Or more importantly, can we find the commonality among this diversity so that we can understand, or we can use it for biomarker analysis? So these are different questions, and what I used this study to work on. So my aim was to use this PDX data to design and implement machine learning models to predict tumor responses to different treatment regimes. And why do we need to do that? So the efficacy or the effectiveness of the cancer drugs is very strongly patient dependent. That is why we are moving more and more towards precision oncology and personalized medicine, because on an average, only 25% of the cancer patients actually respond to cancer drugs. But unlike your normal painkillers, you will have 80% of the patients responding positively to the painkillers. So currently there is a need to predict which patient will respond positively to the treatment. And if not, there is no need to spend time and energy to treat the patients with the treatment. So for this study, one obviously cannot use patient data because of the difficulty in the uniformity among the patient data, but we can actually use these mouse model data to predict the treatment response. So how did I do that? The gene expression data, the copy number variation data, and the gene mutation data were the features from which the machine learning models defines a function to understand the tumor response. So when this model was presented a test case, it tries to predict the tumor response to that particular drug. So such a study, this model was applied to breast cancer and colorectal cancer PDXs, which were treated for 26 different treatments. So what did we find? traditionally, canonically, single genes were used as biomarkers that predict the onset of cancer. And the same set of single genes were used as a treatment biomarker also. For example, if there is an association of a gene to the onset of cancer, the same gene was studied whether this particular cancer will respond positively or negatively to any given treatment. This is obviously and logically is not a very accurate predictor. So this is not a very successful model, but from a multi-omic data analysis, what we demonstrate is before that I want to introduce you to the accuracy measure that we are measuring the machine learning models accuracy levels. So this is Matthew's correlation coefficient, where an accuracy of 1 would mean the perfect accuracy, like all the test data are perfectly identified or predicted to be how it responded to the treatment. And -1 would be complete opposite. If there is a positive response, the model predicts a negative response. And around 0 would be a random response. So what we want is these data points to move more towards 1, so that we get better accuracy of our machine learning model. So in that one, so when the machine learning model was used to predict on single gene markers, you see the accuracy is not very great. It's actually it's very bad. So when multi-gene models were introduced in place of single gene models, the accuracy is improved, and the accuracy level can further be improved by combining or by including more than one profiles or more than one molecular profile, like gene mutation data combined with gene expression data combined with copy number variation data improves the accuracy tremendously. So to quantify, we actually observed we could predict 19 out of 26 treatment regimes. We could predict correctly 19 out of 26 treatment regimes. So this was what we performed, and what could be done, what could be done more in such a study. So one can extend such an analysis to include patient data from TCGA repository, but that involves extensive curation of the data, but one can still do it. A gene set enrichment analysis, like what Oja presented, of the biomarker genes will throw insight into the biology behind the response of these tumors to different treatment levels. So this is how we did, and such a study was actually performed for selected cancer and for selected treatments. So one can obviously extend this study to all the cancer to find the commonality of these cancer cells to react to different treatment regimes. So these are the different future steps that the team is currently working on, but I finished my part and I joined OmicsLogic. So I hope this project was informative and it does inspire you to utilize curated PDX data. This curated PDX data will be available at the OmicsLogic platform shortly. And this, this platform, as you are aware by now, with different introduction from Elia and from Harpreet and from Alika, provides a very well organized research fellowship program that I want to speak about in the next 2 slides. Let me go to the next slide. So yes, so from my research experience, there are 3 main challenges that all researchers will need to overcome in any research program. And this research program is designed to target specifically to address these. 1, time. So a lot of time is actually spent on learning research methods from multiple sources with a varying level of reliability. But in the end, you are going to use only 1 or 2 methods for your question of interest. So what we have, the learning platform, the OmicsLogic learning platform, provides you with a very well-planned learning modules and coursework. This coursework will prepare you for any challenges you might face with the data analysis and or the biological interpretation of your data analysis and your research question. And another big challenge in doing computational work or bioinformatics work is the presence of, or is rather, there is a limited access to bioinformatics expertise or a reduced availability to bioinformatics experts for the students. So in this program, the research fellows will participate in cutting-edge bioinformatics research led by mentors like me, like Harpreet, and several other mentors. You will find information in the website. We have years of expertise in computational biology, bioinformatics, and also including emerging domains like space omics. So this network of mentors that we have will provide you with expert feedback and peer review from the inception of your topic to the final results. And these results will be worthy to be sharing among the scientific community and to the world. And one final challenge is limited access to infrastructure that is required to perform big data analysis. I think this is the most important challenge that many students face right now. So the cloud-based analytical Tauber bioinformatics platform that you are introduced to today, it provides all the infrastructure that is necessary to perform big data analysis in an intuitive environment with the help of flexible, interactive, and visual pipelines. So the research fellows will get access to this platform and a list of curated public data sets with which you can design a big data analytics pipeline on multi-omics data, or one can perform translational research, or one can address clinical questions, or biomarker identification, or treatment stratification, or variety of questions that you can think about leveraging this data and infrastructure. So who can benefit from such a research fellowship program? So this research fellowship program has been designed to help young research students like you to gain access to the premium resources on bioinformatics for the analysis of complex high throughput data sets that I presented before. And like Dr. Harpreet also presented, and also we observed from OJA also. So, and the experts will help you complete a research study on your own. So if you are an undergraduate or a master's student, you can actually engage in research and you can develop a deeper understanding of the subject matter than that is possible through your classroom routines or classroom instructions alone. So such an early exposure to research has been proven to be very helpful and can be the perfect launchpad for your career. You can also apply the skills. That is one of the most important point. You can apply all the skills practically, the skills that you learned from your classroom instructions, to a project of your own and to the project of your that piques your interest. And more documentation wise, you can fulfill the requirements set by your university syllabus or curriculum. So if you are a research student, so we provide, you'll get an excellent opportunity to work one-on-one with the network of mentors and to use the T-Bio big data infrastructure to be part of your research question, or to be an independent study to develop new skills that you are interested to do that. So with that, I thank you all for your interest and attention. And this concludes my presentation. And I think I would like to pass the stage to Subrajit Barua. He's an astrobiologist and he works on space omics and space health management. It's very exciting to hear from you, Subrajit. Please take over the stage.
Well, thank you so much, Dr. Raghav, and it was lovely hearing to you and your perspective towards bioinformatics, its usage. It was amazing to learn about the...
Sorry, any questions? Yeah, please go ahead.
Sure. So it was amazing to learn, and I hope that our audience also has enjoyed every bit of it. And I hope that everybody is as excited as I am for this particular research opportunity, which we call the research fellowship. Now, quickly, I would just talk about a student club, which is called SciOmics, and what is the agenda of this club, what are we targeting to do, and what would you benefit by being a part of it? Well, hello everyone, and thanks for tuning in to today's webinar. It was amazing to see you all here, meet you virtually, and also learn about such amazing opportunities and research going around. And I'm sure that you are also kind of interested or already starting to hope that what can you do and how can you contribute and how can you also be a part of this exciting community. Well, that's something that I am going to talk about today. So my name is Subrajit Barua, and I am the Scientific Outreach Coordinator at Fine Biotech, and I'm also the president of the SciOmics Student Club. I have an MS in molecular biology and biotechnology from ITMA University, St. Petersburg, Russia, and I am a researcher at the International Laboratory of Solutions Chemistry and Applied Technologies. Well, to begin with, Psyomics have been making an impact all throughout the globe since its inception. We have a huge community of more than 1,000 enthusiasts all over the world. We have been exposed to more than 20 great scientists and their webinars all throughout. We also have a very strong alumni network that consists of more than 30 successful candidates from all over the world who have been a part of OmicsLogic Research Fellowship or Pine Biotech's research internships, and now they are out in the world making an impact, making a difference all throughout the globe. So let me talk a little bit about what Psyomics is all about. And to do that, I would begin with a very simple story of every student because, I have been a student, I am a student even now. So I pretty well recognize every student going through something of this sort. Well, I know that you guys would also agree. And if you can agree to that, do let me know in the chat section that we do get interested in doing a research very often, right? We get some idea, we get to know about some information, we watch some webinars, some presentations, and we are so hyped up to begin our own research journey. Maybe we have a task to do, we have an internship to do, or a project to submit as a part of our college curriculum. So we do get interested in doing a research, but there are certain barriers because of which we are unable to complete it, or even if we complete it, then we are not very satisfied with whatever we get, and we just hope that there would have been something better that we could have done. So what are these barriers? Well, sometimes we are unable to define a proper research question which is of relevance and which would be appreciated by the scientific community, because the scientific community is so dynamic, it keeps on changing every day. So maybe we are not able to define the question as required. Sometimes we face one of the major hurdles while doing any research project is we can't find a good data set, just like Dr. Raghava and also Ilya earlier was talking about, that finding a good data set which is curated and which is worthy of analysis bioinformatically using the various tools and algorithms statistically is a major challenge and a very difficult task. Finally, there can also arise a lack of skill sets which are required in this modern world. Well, we might know a lot of skills, we might have experience in many things, but the world changes so fast. So maybe we need to develop and increase the level of expertise that we have in the current technologies out there in the world. But why a student can't address these issues? Well, there can be various reasons. Some of the possible reasons might be there's no one to guide this particular student. They don't find someone enough close to them who they can express their problem, their situation, and who can be like a mentor to them. Then maybe they have some supervisors in their college or their laboratories, but they're too scared or shy to talk to them and consult them. And maybe they often think that the question that they have or the doubt that they are bearing in their mind is too silly to ask someone. They might not know the places where you can find good quality data for your analysis out there, which are publicly available for anyone to analyze, but we don't really have the knowledge of where to look for the data. Finally, there can be a major issue of lack of motivation. We might have that enthusiasm to begin with a project, but maybe in the long run, we just lose it and we just pile it up somewhere up there in a trunk. Well, these are the reasons or the issues which Psyomics like to address. At Psyomics, we have a huge community of experts and students from all over the world with various levels of experience and backgrounds whom you can think of as your friend, philosopher, guide, whom you can talk to. There's no need to be shy because if you join our community, every day you will find someone posting some questions which might be not very big in a matter of issue, but it's definitely treated with respect by every member of the Psyomics Club, because we have been there, we all have been there, we have all started from level zero. So it's like that particular friend whom you have always been hoping for to arrive in your life, with whom you can talk, with whom you can discuss, get advice, and move on with your research life. Also at Pine Biotech, just as we heard from all the experts today, we have great scientists with loads of experience in academia and industry who are there to guide you and who can be your mentors for making your research project actually take shape. And finally, when you work in a group, when you see others progressing, you also develop that motivation to get to the table to uh, power on your laptop and to get started with your project, to be at par with everyone in that particular community. So that healthy competition atmosphere motivates each one of us to get the best out of ourselves. So here at Psyomics, we would like to help you, first of all, to get a very good research idea or a research question, which is at par with what's happening all around the world right now, which you can get by attending several guest lectures, which would be absolutely free of cost, and they would be twice every month. So you get to attend their lectures, listen to them, get their advices, learn about various opportunities, about research going on around the world, what's the cutting edge situation right now in one particular domain, what's the skill sets required to start your own research project. Then you would get great mentors at Pine Biotech who can help you out with refining your question, with refining your idea, and also helping you out with the various skills or the tools that you need to perform this particular research, the methods. Finally, you can obviously talk to other peers who are part of this club, discuss problems, clarify your doubts, talk about certain errors that you might be coming across in your particular research project, or talk about new challenges which you can solve together. So that's a place where it happens, where you get new ideas. And finally, you would also get access to various training programs which would be launched by Pine Biotech in the near future, which you can get enrolled in to develop new skill sets that are very much prominent in today's world. So briefly, I would like to talk about what are the various areas of interests to us, the club members, as well as to our mentors. Well, for example, the buzzword cancer, we are interested in learning how bioinformatics is changing the way we treat cancer, the way cancer research is progressing, and bioinformatics is at the core of those sort of research. So that is definitely our number one interest. We are also very much interested in looking at various metabolic diseases like diabetes, and also we are interested in knowing and learning the role of bioinformatics in treating infectious diseases like COVID-19 and ultimately vaccine development. Because as you might know, the rapid development of vaccines all around the world was facilitated with the help of bioinformatics, especially. And there's lots more that's going on and that interests us. Many of them are very new to this domain of bioinformatics and omics. For example, personalized medicine, although not so new, but it's something which we are beginning to get a grasp of at a very young level of our career. So that's really another exciting domain which we would love to learn more from the guests and from the experts and the mentors that we have. Cheminformatics, that is a use of bioinformatics to develop drug targets and drug candidates with the help of molecular docking and other tools. That's also something which really interests us. Then comes my favorite area of research, which is astrobiology, which specifically is looking for molecular signatures or biological signatures throughout the universe, not only limited by the conditions on Earth, but also beyond. And not only that, but also to analyze the effects of space flight, the hazardous environment on the molecular level of the astronauts, and to actually leverage bioinformatics to understand what's going on in their bodies, in the cells, and to use that knowledge to transform the healthcare system on Earth itself. There are numerous examples of how that is being done, but we will talk about that maybe on one of the Psyomics Club sessions. So definitely do consider joining the club. Then there's agrobiotech and much, much more that's going on. But the most exciting part, which you would get exposed to by being a part of Psyomics Club and also by being in close contact with Pine Biotech, is something which is known as faculty-led projects. So Pine Biotech has a lot of collaborations going on with universities and other organizations all around the world. And every university or organization have a big number of faculties who are interested in one particular domain of their research. They have been working on that domain for years. They have done their PhDs, their postdocs, and many years of research in those particular areas. Now, what you get as a benefit by being a part of this community is sometimes it might so happen that you do not have a research idea or a question of your own yet, because you are starting out right now. You are in the first or the second year or the third year of your undergrad, and you're really getting exposed to bioinformatics for the first time. That's pretty normal. I have been at that place myself, even. So, what's the best way to get started at that particular level is to get or to join a team of some great faculty all around the world who are working on their projects, and you just get to stand with them and look at their projects, look at the methods that they're using, look at how they're addressing the various challenges, how they're shaping their question, and you get to learn a lot by doing that. Now, as you might understand, it's often not possible to contact those faculties, to go to their organization, to go to their places. The only time you can do that is when you go for a research internship or something like that. And that's also for a short period of time, like 3 months. But the major advantage that you get by being a part of Psiomics and also by staying in close contact with Pine Biotech is you get exposed to such faculty-led project opportunities all throughout the world, not only in your country, but also beyond. Now you can participate in their research teams, get to work closely with them, attend team meetings, get to know the ways and the strategies they are developing to address the challenges and the way they are formulating their research project. And I find this particular opportunity really exciting because it can transform your entire career of bioinformatics research in the future. So how would specifically our mentors and the courses which Ilya showed you in the beginning of the webinar that are already listed in the portal help you with your research project and to really get started with your research journey? Well, they would help you to put together all the pieces of the puzzle. To do a research is a big puzzle because there are various pieces like literature review, finding a good data set, finding a or defining a good research question, then bringing together all the required methods, interpreting the results, and coming up with a conclusion. These are all pieces of a puzzle. But if they can be put together in a logical way, they would look a beautiful picture like we see in a jigsaw puzzle. So that expertise, that guidance to do that will be provided by our mentors and all the asynchronous courses that you can go through yourself, and also give you a direction in which you can head in the future, where you could get a master degree or a PhD or maybe a job opportunity in the future. All of this guidance and these information would be provided by our mentors, and you would get that if you are a part of the Psiomics Students Club. Now, this model of whatever I said has been tested and proven by various students, but today I would like to share my experience or my journey in brief, that how I benefited personally from this sort of model. So how I started learning bioinformatics as a young student who's doing his undergrad and knows nothing about bioinformatics, heard the term for the first time literally, and ending up becoming a spaceomics researcher, which is completely a niche domain, which is practically very new and very few people are working in this particular domain. So as you might have guessed, I was also a research intern at Pine Biotech. That's where I got baptized in bioinformatics, got to know about the various tools, got to know about what is data science in biology, how a curated data set looks like, how we can use the various pipelines, the various tools, interpret the results, formulate a research question, give shape to a research project. Everything I learned here. So you can see that was my certificate of the internship that I did. And here are some other certificates that I earned by going through the various tutorials out there in the Pine Biotech portal. So this is a place where I got all the necessary information to begin with. And then one fine day, I was looking through television and various other blogs. So as you might have already known, you guys have also come across various shows and such materials in Discovery or any other channels that astronauts face a lot of problems when they embark on a space mission, when they go to space. And that's all because of microgravity, radiation, isolation, etc., etc., like bone and muscle density loss, physical strain, cancer, gut microbiome changes, etc., etc. But that's not where I stopped. I took one step further to translate this information or this need for looking into this particular domain to convert it into my research interest and to convert it into my research project. So that's where again I got connected with Pine Biotech. I approached them with my idea, and they were very happy to welcome this idea. Although it was new and a very niche domain, they helped me all throughout the journey. The excellent mentors at Pine Biotech helped me at every stage, helped me address every challenge that I faced. And finally, I could devise some result out of the research question that I had, which gets aligned to NASA's human research program, that is basically to identify and to address the challenges faced by human beings inside the space conditions. So as you might see, some of those research are already in a preprint condition right now, and I'm hoping that they would get published. But that's not where I stop, because this is just the beginning. Now I am looking towards other data sets, more complex data sets, more complex organisms to look into, and more complex pathways and biological processes which are out there to be analyzed. These data sets are available in the public domain. What they require is the proper interest, the proper motivation, and the proper guidance to analyze and get something very meaningful and outful, like useful from those particular data sets. And that's how being a part of this rich community helps you. That's how much the being a part of the community can elevate your ideologies and your experiences. And as you might have already guessed, this is where everybody of us once have been, the stage where we get an idea, and this is where we want to be. We want to be successful, we want to make an impact, we want to announce ourselves into this domain that, yeah, we have also done something. But there's a big gap between those two areas, and that's the gap which Psyomics loves to bridge, and that's our vision, to provide you with the necessary amount of guidance, the necessary amount of resources, information, and tools to go from here to here and make an impact into the world. So, in a nutshell, the agenda of Psyomics looks like this. We would help you to learn about the current research standards and challenges present out there in various domains of interests all throughout. We would also help you and provide access to various new skills, and also our mentors would help you define impactful research questions. And finally, our community would also help you to interpret results, publish in various journals, or present it in some great conferences all throughout. How do we aim to provide you the information? Well, as I said, by inviting guests who have lots of experience from all over the world in various domains for a webinar, which you can attend for free. And here's what the timeline, which is quite tentative, but still is in our action of plan. So in November, we have the theme of cancer. So we will be having two great speakers talk about cancer, their research opportunities. In December, we would have infectious diseases. In January, we will talk about astrobiology. In February, metagenomics, microbiome. And March, personalized medicine, and so on and so forth. It will go on and on without stop. And every month, there would be a new theme, a new particular domain of science and bioinformatics, which would be addressed, talked about, and all of you are welcome to be a part of it. So if all of this sounds exciting and good to you, do consider joining Psyomics today. It's absolutely free of cost. We do not charge anything to be a part of Psyomics and to get access to the community as well as the various guest lectures. So you can join us through WhatsApp, so through Telegram or Slack channels. The details will be shared. You can also find all those details on social media platforms of Pine Biotech or my social media platform. So if you want to stay in touch with me, get in touch with me via LinkedIn or Instagram or Twitter, and I would love to talk to you. And if you guys have any questions, feel free to put them in the chat. I would love to address them. And finally, hope to see you in the next Psyomics sessions. Good luck with your research journey, and I wish all of you all the best. So right now, I would pass it on to Sonalika to take the session further. Thank you so much for listening to me today.
Thank you so much, Shubhroji, for sharing with us about the Psiomics Club. I'm sure the club is doing some amazing work. And I've also shared the link of the Slack group with you all. So this is what the Slack group looks like. And if you have registered yourself on the program page, then you are going to get the invitation to be a part of the Slack channel automatically. So all you need to do is just click on the link and you will be redirected to the Slack channel. Also, we have a Skype group, the details of which has already been shared in the chat box. So now I'll be just taking you all with the registration process, and also many of you asked about these scholarships. So let's now discuss how you proceed further to enroll yourself for this very exciting research fellowship program and get started with your research project. So once you visit this research fellowship program page, you are going to get all the details that you are supposed to know for the program. And today we discussed these 3 steps in greater detail. That is, you are going to get ready through training and then find an impactful research topic and present your analysis for expert feedback. The 3 major outcomes of the research fellowship program could be either you could come up with a literature review or present your research work in a poster presentation competition maybe. And the third one is definitely publishing your research work in reputed journals. And here on the program page itself, you will get the details of all the mentors. You heard from some of the mentors today. And also you could register yourself in the form here, which is mentioned in the program page. And once you register yourself, you are going to get all the details about how you can further enroll yourself for the program and how to get started. Now we do understand the need to make this program reasonable for students, along with the basic requirement of sustaining the program altogether. So we have designed this new program page for the computational biology internship program or the fellowship program. So don't get confused between these 2 terms. At many places for the term internship, fellowship word is being used. So you could find the basic structure of the research fellowship program here on the page itself. How do we proceed further? So first of all, you all would be supposed to go through a course material and then come up with your research proposal, submit your research question, and then get started with your research project. And how do we meet? We are going to meet twice every week to take up your doubts and see your progress and also suggest you or recommend you the further steps that should be followed to carry out your project. And you can go through the structure here on the program page, which is very well defined. And to join the research fellowship program, you can take up the license here on the page itself. So we have 3 licenses. The first one is beginners, the second one is intermediate, and the third one is advanced. Now, these 3 levels are based on the different types of features it includes. So the beginner level does not include the one-on-one mentorship. That is that you could take up your doubts in group sessions. And also many a times participants are not able to take up their doubts in a group session, maybe because they're not able to clarify their doubts clearly, or they do require a one-on-one meeting with the mentor. So that could be taken up in the intermediate license. And then for the advanced license, we also have the technical support that is being provided along with one-on-one mentorship. And also we have different types of license on the T-BioInfo platform that Ilya showed you all. So there is a specialization license, an educational license, and a research license. Now this license has a different provisions that it's going to provide you to work with your data. In the educational license, you would be able to use the big data from the various big data repositories, upload the data and work on it. And in a research license, if you are generating your own data or you are from a lab or a PhD student maybe, then you can upload your own data using an FTP account and carry out the analysis. So this is the page where you can just enroll yourself using like taking up any of the license that you would like to take up and enroll. And then on this page itself, you'll also get to know about the various courses and sample projects that you can go through. Now, all the courses that Ilya also showed you as a part of the research fellowship program, all the courses and the example projects would be unlocked for you all. So everything here under the courses tab, you can go through and learn from. Now, there is one more aspect that I would like to share with you all, that is the scholarship application. So we do have a monthly module of license that you all can take up and be a part of the program. So the module that I just showed you, that is a monthly module. That is, you have to take up this monthly license and carry out your research work. But if you would like to take up the license for a 3 months research fellowship program or a 6 months program altogether, then you are eligible to take up or apply for a scholarship application as well. And this is the scholarship application form that I will be just sharing in the chat box. Once you fill this scholarship form, we will review your application and get back to you to tell you how much scholarship you're going to get for the research fellowship program. And that's the way you can get yourself enrolled. Now, I would like to leave the stage open for everyone to take the doubts. Please put in the doubts in the chat section, or you can also raise your hand and unmute yourself to take up your doubt.
Sonalika, I just wanted to clarify on a few things that were just mentioned. And hello, everyone. Myself, Mohit Mazumdar. I'm one of the co-mentors uh, uh, and representatives who are working on collaborations. Today, I wanted to discuss few things with all of you. So we have some of the exciting collaborations that we recently did, signing some of the MOUs with some of the university departments who are involved in research, some of the departments who are involved in training, and also industry, where we have people from different pharma industry and the biotechnology industry to help students guide through. So about the licensing, which Sonalika was mentioning, it's actually a subscription. So when you're taking a monthly subscription, you're paying monthly. These can be done in 2 months, 3 months, and 6 months. The structure works for different models. So if you are thinking about taking a 2 month research fellowship program, you can go for either a beginner subscription or a intermediate subscription or an advanced subscription. So obviously these are different subscription levels that are available for you to take by monthly if you choose to, take the mentorship and take all the resources or the premium resources and develop a project. But obviously you can start from a very beginner's level that would not require you to take any license, not require you to pay anything. Just join the OmicsLogic portal, and then you can also take help from Psyomics group, as mentioned by Shubhrajit, that would help you develop such research question. And then you can take the decision to go for big data analysis. So one of the reasons that there is a subscription to this program is because that we are actually providing you with a platform to do big data analysis. And that platform itself would require us to, give you a lot of space on cloud, as well as access to all the resources that we have developed. So that's why there's a subscription. And coming back to the question that Crystal has, that she's saying, I'm Brazilian and have no money. So that's perfectly fine. We have different types of scholarships that are available for different kind of responsibilities. So Crystal, we also have a research campus ambassador program, which you can join. And one of the basic objectives of that program is that you bring awareness about bioinformatics, about OmicsLogic training in your area. And we kind of get that in a form of a report. And then we provide you a full scholarship to do this program for 3 months or 6 months. So that is also a way to do this program and get supported, but that would require you to give a lot of time and effort and learning and applying all of this. So the best way to develop a project is obviously to work directly with the mentor, uh, do the research webinars. I mean, come and join the research webinars weekly and discuss your problem. So we are available 24/7. And then there are like online courses and the portal that you just saw, which is available 24/7. So all of this actually combines to become a OmicsLogic program or a research fellowship program. And I just wanted to quickly share with you about some of the university programs that we have. So we have collaborated with MIT University in Jaipur and in Noida. And if you are a student there, you can reach out to us and you can let us know, and we will be able to help you with registration relevant to the university. So just to tell you about the 3 different collaborations and the collaboration with industry. So we recently, just a second. So we recently collaborated with MIT University Jaipur, and there's a reason somehow I'm not being able to share the full screen. And we discussed about different multi-omics courses and integration of, integration of our programs into their internships. So students will be joining from MIT University Jaipur today. Over 130 people joined, and we discussed about the entire research fellowship program and development of projects in group. So one of the way to make it sustainable is to also work on group project or collaborative project. And all of the things that we have discussed today and all of the things that we've been able to achieve is because of the collaborations, is because of that we are working as a team. So one of the biggest aspect of this kind of training and this kind of community is about collaboration, is about understanding each other, is about doing these tasks by dividing the tasks and by showcasing our own expertise in different areas. So similarly, we have a collaboration done with CGC Landron, Chandigarh. And then we have launched the research fellowship program for the students there. And there also we are working with Department of Biotechnology, Microbiology, and pharmacy to help students develop research projects in those areas and work through these different resources. And that all starts from the basic training. That all starts from like going through those basics that has been reiterated over and over. And then we do have another collaboration with MIT University Noida, which turned out to be like a historic kind of a collaboration because it involved several departments. So we are talking about at least 6 to 8 departments within MIT University Uttar Pradesh, and then defining of those research questions during that several meetings that we had. So we had brainstorming sessions where we discussed that how this can be integrated, how the expertise from faculty can be integrated into a program where we can help students, giving them a holistic understanding of how to work on or define a research project and then work with a faculty-led research project, as Shubhrajit was telling in his presentation. So these are some of the opportunities that are there with universities and those universities who have reached out to us. And obviously, we are working on different type of projects within those universities. Along with that, one major collaboration that I would wanted to, showcase to you all is the collaboration with industry, which is with FABA Academy, which we recently did. And in this meeting, we discussed about how we can help, how industry can help in recruitment, how industry can help in giving job opportunities. And we met with people here, as you can see on the screen, that are leaders in industry, leaders in biotechnology. They are represented in big pharma companies, and they're also working with AI and drug discovery. So one of the objective of this collaboration is to be able to develop resources in data science and AI in healthcare. And I'll be working very closely with the group of people here, developing technologies and resources for students, and also giving students an opportunity to work on projects related to AI in drug discovery, where we are thinking about small molecule screening, in using information from protein folding, structure prediction using AlphaFold that you must have heard about. That was recently came out and surprised the structural biology world, and not only the structural biology, but maybe the entire world. So another application, data science and AI in clinical trials. So this is a huge also when it comes to AI-based drug discovery. So what we are trying to do and what we are trying to do in this program would be helping you decide on some of the important research areas first, and then go to another step to work and develop the research project. So if you are not sure like which research area that you are more interested about, then you can go about the intermediate training after doing the basics that would tell you about these different research areas such as precision medicine, population health, agriculture and biotechnology, space omics that we discuss in length. So you'll be able to work on 21st century problems, and I'll be available to help you as a mentor. So I have expertise and over 40 publications in different research, addressing different research questions using bioinformatics. So please feel free to reach out to me and reach out to our team, and we'll be able to help you go through and complete this journey. And you already saw the subscription levels for the research fellowship program. You know about the SciOmics program, and it will be great if you also can become the ambassador of these programs and help others who can be a good fit for this. So with that, I would like to conclude today's session, and it was really, really great to see you all. And I'd really like to thank Dr. Raghavendran, Dr. Harpreet, Ilya, and Sonalik and Subhrajit for giving us wonderful lectures throughout this way. And we will be looking forward to meet you next week. So next week also we have a wonderful session coming up, and we'll be emailing you about that on different OmicsLogic programs that we just discussed. So we'll be talking about infectious diseases in much more detail. We'll talk about metagenomics, we'll talk about genomics, we will talk about transcriptomics, and these specialization programs are how they are helpful in developing and in understanding and finding those research questions. So thank you.
Thank you so much, Dr. Mohit. Thank you so much, everyone, for being a part of the webinar and making it such a wonderful event today. And if you have not registered yourself, then do register so that you get the links to all the groups that you can join and stay updated about the program. Thank you, everyone. We'll be meeting soon. Bye-bye.